Hi everybody, my name is Alex and I'm in George's lab, and I'll be talking about our efforts and the field's efforts in general to produce human organoids and stem cell derived cells which can be used to test these large synthetic genomes. So the way I think about it is there are two broad approaches of doing this. One is a bottom-up approach starting from constituent cells and you build it up, and another one is kind of a top-down approach where you recapitulate development and build large organoids. So in the first approach, in this bottom-up approach, one might start with human stem cells and differentiate them into homogeneous populations of specific cell types of interest. And then afterwards assemble them either by 3D printing methods or organ-on-chip methods and so on to get the desired tissue type. In the complementary approach, we might recapitulate development closely and try to do as much as we can to allow the stem cells to do what they do and derive organoids from them. And of course, these organoids are not perfect. They have high resemblance to real tissues and organ-like structures, but there's still missing parts to them. And so we can use organoid editing techniques to try to enhance the similarity to real tissues. And I'll talk about both aspects in this brief talk. For the first approach, the bottom-up approach, I mean, it's really a daunting task to develop methods and recipes to make these cell types. I mean, Liam just talked a lot about the difficulties in that, so thank you for that. And if we think about how many different cell types there are in the human body, there are probably thousands. And when I say probably, it's because we actually don't know how many there are. I mean, in the human brain alone, there are probably hundreds of different types of neurons. In the hematopoietic lineage, there are lots of variety as well. And all these organ types, we still don't really have good recipes to make them. So this is the major challenge, and there are a lot of labs that are doing amazing work in the stem cell field deriving these recipes, and we're approaching it from a different way. We were thinking more in terms of what defines a cell type. Well, that's probably a transcriptional network that underlies its identity, and what controls it, those are probably transcription factors. So we took kind of a naive approach and said, let's screen all the transcription factors in the human genome and test which ones can make these cell types. And unlike other labs, we are not focused on a particular cell type. We're focused on trying to make as many of these cell types, as many of these thousands of cell types as possible. So to do this, we assembled what we call the human TF-ome, the collection of basically all canonical human transcription factors, over 1,600 of them. And we threw them into iPS cells and just asked the question, which ones can encourage them to differentiate into basically any differentiated cell type. And about 75% of this collection is derived from cDNAs originally. We had a lot of help from Mark Vidal's lab and the Orpheum Consortium, which contributed to this collection, as well as others. And for the rest that we couldn't get our hands on very easily, we actually synthesized about 25% of this collection from the synthesis company Gen 9. So with this transcription factor collection, the TF-ome in hand, we tested which ones can differentiate stem cells. And in the first pass, with no combinations, just single TFs in human stem cells, we find about 80 new recipes to make cell types. So this is a large number from one study alone. Typically, you would focus on one cell type for 1 or 2 grad students, PhDs. Here we've putatively made about 80 new recipes, and we've recovered a lot of known developmental transcription factors as well as many novel ones as well. So for the next few minutes, I'll talk about some of the specific cell types we've been able to make. So in this particular example, we found several recipes, this is just one of them, to make a homogeneous population of human neurons extremely rapidly in 4 days with greater than 95% purity in basically a synthetic approach, right? We're not recapitulating development through progenitors and ectoderm, et cetera. We're going directly into the cell type of interest very quickly, very efficiently. So here is going to be a movie of starting as a stem cell colony, a nice compact stem cell colony, and we quickly induce expression of this transcription factor, and immediately the stem cells differentiate and leave these colonies and migrate out of them. And over this time course, they start developing mature characteristics of human neurons, extending axon and dendrites, and we can further convincingly show that they express mature markers of neurons. We can have global signatures from transcriptomic data and functional data from electrophysiology that indeed they are electrically active and they fire. So this is one example, and we have multiple examples of different types of neurons we're making. And another example, well, now we're able to make human blood vessel cells extremely quickly in 4 days. Again, skipping all the progenitors, going right into the cell type of interest. So this is 2 days into differentiation. You can watch that there are stem cells that are leaving stem cell colonies and developing into this characteristic morphology of blood vessels, very flat, space-filling, sealing morphologies, to seal this blood vessel. And this is an earlier time-lapse movie where we were optimizing the protocols, and since then we've basically developed, again, greater than 90% efficiency at deriving the cell type in 4 days. And again, we can look at mature markers, express mature markers, have global signatures of blood vessel endothelial cells, and they're also functional in that if you have the right matrix, they can form blood vessel tubes in a matrix. So with all these new recipes, again, just with singles, now we're starting with combinations as well. With these singles, we can even think of a cell chip assay similar to a SNP chip, where you have a chip, you have these transcription factors that are ready to go, and when you want to test your new cell that has a new synthetic genome, you can put it on this chip and differentiate them into a number of different cell types and ask questions like, if these are stem cells, are they affecting pluripotency? If you have another spot that makes them into endothelial cells, do they have any leakages in capillaries? Are they functioning properly? Neurons, are they functioning properly in terms of signal processing, and so on? And we're beginning to do these type of things where we're using microfluidic technologies to pattern these transcription factor cocktails and really get this and bring this into a reality. Just for the last few minutes, I want to talk about the efforts that we're pushing into for human organoids. I mean, this is a very rapidly progressing field, and in this case, you might imagine that you have a synthetic genome, you test on a cell chip, for example, it passes all those criteria. Then you want to move into more complex assays, more laborious assays, where you go into a suspension culture and then you push it several weeks further into these different organoids. The efforts here that we've been pushing for is trying to make bigger organoids, because these organoids are typically 1 to 2 millimeters, largely because they lack vasculature. Their interiors are apoptotic, they're not receiving nutrients, so you really can't get real organs. You really have organoids that are millimeter scale. So to really push that towards an organ-sized organoid, we are using organoid editing techniques and adding vasculature in them, and from very preliminary studies already, we're already making bigger organoids with these synthetic blood vessels. So I just want to summarize very quickly the things that we've been able to do in terms of enabling technologies and building cell types. This TFOM library has generated a lot of new recipes. These might be useful in a cell chip type assay. And these organoids we're really pushing these organoids towards bigger organoids with more complex morphologies. I'd like to acknowledge the people who helped on this project, and I'm happy to take questions.
So far, it's just single transcription factors. Yep. And so if you looked into combinations, I see you got no combos there. But
very explicit. Yeah, everybody asked that. Yeah, so we're pushing that angle quite quickly. Of course, we can do some unbiased search, but the combinatorial space gets enormous. 1,000 times choose 2 is like a million, and it just becomes very difficult. So we're using more targeted approaches in multiplex to test thousands of combinations at the same time. So that's an ongoing project right now.
Touched on this, I might have missed it. Do you think you can create radically new cell types that we really don't have in the human body at all?
I think we're definitely making a lot of that. I mean, that's like kind of the other face of the coin. I know. Well, a lot of these partially differentiated or partially reprogrammed cells, some of them are on the trajectory towards the correct cell type, but probably a lot of them are heading towards a attractor state that might not exist. Exactly, yeah, so we're very interested in seeing synthetic cell types that might be useful as well. Yeah.
Hi everyone, my name is Luhan. I'm the postdoc from Church Lab, also the co-founder together with George for the startup eGenesis. Today I'm happy to present to you our progress of using mammalian genome engineering to deliver xenotransplantation as a safe and life-saving procedure. So here we are looking at the number of the patients suffer from the end-stage organ diseases in US. So there are over 2 million patients suffer from the organ failure. Only about 123,000 of them are putting on the waiting list. And only 1/10 of them can get transplantation each year. So there's a huge delta, and human life is at stake. The situation is worse in other parts of the world, especially in Asia, when there is no donation culture. So hypothetically, there are probably 3 ways to solve this problem. 1, we have more donation. Second, probably as Alex introduced, we can use stem cell to produce an organ. And the approach we take, which we hope can help us get there faster and quicker, is to utilize the animal as a bioreactor and change in the way that human compatible. So the process works in the way that we start by engineering the somatic cell. Then as people clone Dolly, we clone transgenic pig from the engineered cell, then deliver organ to the patient. We didn't invent this idea, so people have pursuing this for more than 2 decades, and big pharmas invested very heavily in the 1990s, but it's a huge failure. And there are 2 reasons why it didn't work. First, as you can imagine, there are huge rejection issues between the pig organ and human body, and people underestimate how many antigens they have to remove, and there was no tool to do that. Second, people identify the endogenous retrovirus reside in the pig genome, and if you culture a pig cell together with a human cell, the virus will jump from the pig cell to the human genome. So it didn't work, potentially can become a plague. And that's where we think CRISPR and other advancements in mammalian genome engineering can come to play. And our lab was one of the first labs to demonstrate we can use CRISPR to engineer mammalian genome. We think the beauty of CRISPR is the multiplexability. With such a tool, we can conduct high-throughput engineering to remove the antigen, introduce the immune inhibitory factor to confirm the pig organ human immune compatibility. In addition, we can use the tool to eradicate the virus from the porcine genome to ultimately address the safety concern. So this is a study we published last year. We first utilized a sequencing technique to identify there are 62 copies of the endogenous retrovirus, in particular in the genome of particular cell line we are working at, and we designed CRISPR specifically to tackle the catalytic domain of the reverse transcriptase of the virus genome. And after the treatment, we got the cell line with 100% knocking out of those 62 copies of the virus. And here we are showing that if we culture the pig cell together with a human cell without modification, they are still prevalent infectivity, whereas if we do that with our engineered cell line, there were minimum infectivity compared to similar to the negative control. So our work increased the multiplexability of gene editing in mammalian cell by almost 2 orders of magnitude. And more importantly, we demonstrate we can solve the biggest safety issue of xenotransplantation. After that, we move to our second challenge. How can we engineer the pig to have the immunology feature? And over the last 1 decade, there are some scientific progress that people produce different transgenic pig, and each one of them carries some modification. The good news is all the pigs are viable, and they all shows beneficial effect for xenotransplantation. But it takes years for them to generate those pigs, and some of the modification couldn't be implemented due to the lack of the tool. So over the last few months, we have generated pig 2.0 cells with 12 modifications, which we think can address antibody binding, complement coagulation, as well as the T-cell toxicity issue. And here is some preliminary data. We show that with modified pig 2.0, we have dramatically decreased human serine binding affinity by 80%. And we also conduct the whole genome-wide screening. We designed CRISPR, 70,000 CRISPR to target every gene in the pig genome 3 times per gene. And having such a library, we are trying to identify the novel target, which can help us fill the gap between the performance of our current cell line and the negative control. In the meanwhile, with the help of the Denmark Aarhus University, we built the somatic cell nuclear transfer system in which we can take the engineered cell, put into the pig egg, and get a pig blastocyst in 1 week, which we are going to do the embryo transfer to get the pig, as well as also a very quick feedback to let us know what's the developmental impact of our modification on pig. So we are in the process of generating our pig 2.0, and every cell, every tissue, every organ of our pig will have the identical genotype. So we think our pig can serve as a bioreactor to produce different organs different tissue, such as for heart transplant, liver transplant, pancreatic transplant, as well as neuronal transplant. And I hope I convince you that the advancement of gene editing really reinvigorate the field of xenotransplantation. In the meanwhile, I think if we can solve the immunology issue, we can do things beyond that. We can engineer the liver to be cancer resistant, as Jeff mentioned, virus resistance, as George mentioned, will produce some non-essential amino acid, as Harris mentioned, and we don't need to worry about the ethics issue if we need to use that directly on human being. So to a certain extent, xenotransplantation open a back door for us. We can really exercise a tool to push the envelope of gene editing in terms of the scale and complexity, which may have the impact beyond our practice. So last, I'd like to thank George for being a visionary and enable everything happen. And my team, specifically Mark Guile, led all the synthetic biology as well as bioinformatic effort, don't build the embryogenesis system, and working together with Elan to lead the effort of the PIC 2.0. And I'm happy to take any question. Scott.
So in the paper where you described the inactivation of 62 loci at the same time, these were all PERV loci. Do they include all the PERV families, the C and the D, and I think there's an A, and do they hit all of them? And then in the cell type that you used to achieve this, is that clonable, that cancer cell line PK15? Because I'd be interested if it is, obviously.
So Scott asked two questions. First, whether we hit all the PERV family, and second, whether our cell line is clonable. The first one is yes, there are A, B, C, and A-C family, and we hit all of them. And there are some pseudogene, and we designed the CRISPR in the way that we only target those active PERV, not touch other part to prevent some genomic instability. And the second question, no, the cell is not clonable, but we are repeating our process in the clonable cell line and hopefully can produce a transgenic pig with such a feature.
Hi, Liam Holt from NYU. So I was wondering about the immune rejection problem, and I think you were talking about changing the antigenic surface of the cell, but also cells present peptides from the interior of the cell. I was wondering if you could break that pathway so that you aren't getting arbitrary pig peptides displayed.
Yeah, so Mian's question is, even you can remove the antigen from the pig surface, the cell debrief will be picked up by the antigen presenting cell by the host, then trigger the indirect rejection. And to solve such an issue, we are not only knocking out the antigen, we are inserting some of the immune inhibitor factor, for example, CTLA-IG, which can suppress the T-cell reaction and hopefully can mitigate such a problem. Okay, thank you.
So Vector as a platform for human genome synthesis project. What is human artificial chromosome? This is extra mini chromosome with size 210 megabase in size, pure kinetochore, single copy per cell, mitotically stable, never integrate into chromosome, and hug-based vectors have unlimited cloning capacity. So there is a common way to generate human artificial chromosome is a bottom-up approach based on transfection of human cells by alpha satellite DNA isolated from human centromere. So Hunt Willard demonstrated many years ago that a large block of alpha satellite DNA this is a pointer. Okay, demonstrated that a large block of alpha satellite DNA isolated from human centromere, when transfected to human cells, can form de novo kinetochore, and this kinetochore may be propagated stably during many generations. We designed, so we start to work with synthetic alpha satellite DNA, and this is a principle how we can assemble large block of alpha satellite DNA for de novo Hak formation from dimer of alpha satellite DNA 340 kb. Rolling circle amplification and assembling in yeast by recombination, in vivo recombination. Advantage, each nucleotide may be mutated before amplification. More importantly, we can introduce site for recognition of protein, that so repressor or lock operator, and amplify, and so use this assay for array for Hak formation. This slide shows Hak generated from alpha satellite synthetic array with known structure, so containing TET operator sequence. Hak segregate accurately during mitosis and mitotically stable. This is the structure of the Hak as any other Hak generated by de novo formation. Input DNA so amplified, and we have approximately 1 megabase amplified region. So this Hak contained 3,000 copy of TET operator sequence. We can target these sequences TET repressor. TET or Hak has multiple applications, including study organization of kinetochore, Hak as a vector for gene delivery, Hak to measure chromosome instability, and Hak for synthetic biology. I will give you several examples. We constructed dozen and dozen TET repressive fusion with different protein in order to target human kinetochore and TET or Hak. I will so talk about today what is important for us, VP16 transcription activator fusion with this protein. We demonstrated that expression or binding of VP16 to TET or Hak kinetochore immediately disrupt kinetochore and result to Hak loss. So it means that we can regulate so kinetochore in this Hak. This is very important for Hak when we use as a vector. Hak as a gene delivery system. This is the only Hak with conditional centromere, which we can regulate it, and as other Hak have no unlimited cloning capacity. To convert this Hak into vector, so we introduce unique LOXP site into Hak, homologous recombination in DT40 cells. This is a long story, but so finally we have human artificial chromosome propagated in CHO cells. In these cells, hamster cells, we can introduce any genes with size up to 100, 200 kb into Hak by Cre-Lox recombination. So if necessary, multiple integration is impossible in the Hak. This system was developed Nicholas Lee in our lab. So and system is used combination of Cre, FBT1, FTC recombination integrase. So system is cyclic, and so it allows assembly of large genomic region, unlimited number of cycles. We can introduce in the same locus into Hak. As a proof of principle, so several human genes, including BRCA1, were loaded into human artificial chromosome propagated in CHO cells. So transfer using microcell-mediated chromosome transfer procedure to deficient human cells. We prove complementation. For BRCA1, we identified even new function using this system. And we have good control. We can eliminate Hak by inactivation kinetochore. We have original deficient cells. So now Hak about quantitative measurement of chromosome instability. There is no method to quantify chromosome instability in human cell, but it's very, very important for many reasons. And this is very, very simple test. We have human artificial chromosome containing GFP, fluorescent protein, and of course cells which contain Hak will exhibit green fluorescence. Cells which lost will do not. It may be so measured simply by simple flow cytometry. This system is were developed to check drugs which affect for chromosome instability. Why is it important? Because recently, right now, so chromosome instability is recognized as a major target for cancer therapy. We have to increase chromosome instability in cancer cell in order to kill specifically cancer cells. We did such screen using our test. 100 approximately drugs currently used for cancer therapy in clinics were ranked for frequency of chromosome loss. So highest effect was identified for Taxol and gemcitabine. Treatment result was 60% of cells after treatment lost HAC and of course lost other chromosome. These drugs unfortunately not new, but were developed, introduced in clinics 30 years ago. So we can also work for identification of new genes, collaboration with Phil Hitter lab, using the same system. And now HAC for synthetic biology. So we have two projects. I will briefly talk about assembly of rDNA array to form nuclear organization center on chromosome 21. I remember that there are 5 clusters of rDNA genes in human acrocentric chromosome. This is a large region, from half to 1 megabase, and this is a gaps till now in human, current human genome project. So problem was with cloning, in human genome project, number 1. Right now we are working for isolation of genomic fragment corresponding to individual rDNA cluster and close last gap in human genome, and is use this clone to assemble entire nuclear organization region in the HAC using multi-integrase system. And so cloning is TAR cloning procedure developed in our lab 2 decades ago. And future direction close remaining gaps, assemble chromosome 21 cluster in the HAC, use rDNA HAC model to clarify how present additional nuclear organization region affect cell physiology. Last slide, this is a main conclusion. Conclusion that HAC may be a good platform for synthetic biology, physically characterize copy number control of copy number, stable propagation in different cells, unlimited cloning capacity, opportunity to eliminate HAC by inactivation of its kinetochore, unlimited number of gene insertion, opportunity to transfer from one to another type of cells using MMCT. This is a list, participant, my lab, and my main collaborator, Bill and Shaw, and Hiroshi Masumoto. Thank you for attention.
It's great work. So is there any demonstration of the ability to transmit these by sexual reproduction, or are they stable in meiosis 1 or 2?
Yes, what we did, we published recently work for demonstrating that human artificial chromosome is stable in transgenic mice, can be propagated in mice. We also have this is collaboration mostly with Mitsuo Shimura. He did transfer of human artificial chromosome into embryonic human cells. He also, in collaboration with us, he designed new model for IPS reprogramming. So huge block, approximately 120 KB was loaded into BAC. Into HAC, human artificial chromosome, and this block was suggested by new new information about what necessary for cell reprogramming. So it's stable in embryonic cells and stable in mouse, and so can be passed through meiosis. So idea that this system may be used as a platform to assemble large block, synthesize in vitro or isolate it from chromosome, and we can. I forgot to tell that we have there are two different versions of artificial chromosome, circular and linear with telomeres. So its principle is the same.
Hello, I'm Yinan. Just a quick question. How long can you passage a hack in human cells?
Could you please repeat?
Yes. How long can you passage your artificial chromosome in human cells?
So how to transfer, right?
No. What's the stability during passaging?
Stability is very it's mitotic stability of human artificial chromosome approximately. So if we will compare with normal chromosome, approximately 5 to 10 times low only. So this is approximately the same stability as the stability of normal chromosome. But very, very careful, measuring tell that a little bit low, but not dramatically. So it's principally like normal chromosome.
What's the current state of art to transfer such a large chromosome into the mammalian cells? And is there any way we can do that in a high throughput fashion?
So you're talking about transfers of hack?
Yeah, how to deliver?
How to for delivery? Original protocol developed in 1977 by Frank Radl. Microcell mediated chromosome transfer is working till now. Many modification of this procedure was done by Mitsuo Ashimura lab, in Tottori University. This is now this is very, very efficient procedure, and we can we can transfer one hack from one cell to another cell with high efficiency. So this is a recent modification in Mitsuo Ashimura lab, and also we published modification recently, just recently, this procedure. So this is, and we also demonstrated that this procedure is not dangerous. So we have no chromosome rearrangements during microcell-mediated chromosome transfer.
Hi, I'm a postdoc in Pam Silver's group at Harvard Medical. I don't know what his chair is here. I'm the postdoc working on the human artificial chromosomes in the lab. No one else is working on it. We're relatively new to the game, but I'm going to catch you up in 5 minutes. So why are we interested in hacks? There are obviously a diverse amount of commercial and academic interests. Nowadays, it's not so easy to differentiate between the two anymore. But we're really interested in hacks because, as many of you have mentioned, it will really hack into chromosome biology across all organisms, so not just humans, but it will really push the boundary on many different things. And if hacks get too controversial, I might go into lizards and do like dragon artificial chromosomes, but this is the goal, which is to hack chromosome biology. And so what makes a good hack? And in terms of building a synthetic genome, what do we need to do with a hack before we can do that? And there's a way to score hacks for us, and I'm going to go through the criteria. So the first really important thing is segregation. Having a hack that's centromere competent, and this is the core of chromosome biology. If you can't segregate your hack, it doesn't work. And the next thing is compatibility, and this is what gives hacks a leverage above viral plasmids or random pieces of DNA, which is compatibility with the system. And this is a lesson we can learn from yeast and bacterial plasmids and vectors. They work with the system, they come from nature, they are expressed, they are recognized by cells, but in human biology, we haven't breached that barrier that much. So we keep trying to use viruses and things that will be attacked by human cells will be silenced and degraded. And so our objective of the hack is to make something compatible. The next thing is organization. So we don't want to just create a jumble of DNA that you throw in and you don't know what happens to it anymore. You want to know what exactly is in your hack and what it looks like while it's in the cell and if it gets passed on with integrity to the next few cells. It has to be replication competent. If it doesn't replicate, it's not useful. And expression is also super important. If you can't express of your hack, it's not useful again. And I'd say that in the last 2 decades, we've made significant progress on hacks in terms of meeting these criteria. And now for our lab, we really want to hit the last one, which is shuttleability. And what this means is being able to shuttle it, like yeast vectors, into bacteria or yeast and manipulate them, construct them, and then put them into human cells. And this will accelerate development of hacks so quickly and genome synthesis as well, because if you can use the tools we have right now that work really efficiently, building megabases of DNA in yeast and then shuttling it into human cells, that will make it so much more multiplex and high throughput than we could ever achieve right now. So I'm just going to talk about the last 2 decades of hacks and what they look like. And I'm really glad that Vladimir has gone ahead of me, because now you guys are up to speed on hacks. So one way is top-down, like he mentioned. So taking a naturally existing human chromosome, this is the group from Japan. And what they did was that they chopped this up. They removed all of the bits in between the centromere and the telomeres. So all you have are the core and the ends. And this is now about 5 megabases. It's been truncated, and it's in chicken cells. The other way is to go from bottom up. So de novo, taking a small piece of centromeric repeats, for example, and then amplifying that and throwing it into human cells, and hopefully getting something that exists, like persists as a hack. And so this is what Vladimir's group has done. And both of these hacks are now in CHO cells, where you can load genes on via recombination, and then use microcell MMCT to transfer those chromosomes into your cell line of interest. So this process is still in development, as has been discussed. But we're trying to sort of go back to square one and see if we can make more well-defined hacks maybe, or make it a faster, more efficient process. So while Vladimir has sort of gone through his illustrious career of making hacks, I'm going to talk about our modest beginnings of making hacks. I've been about a postdoc for a year and a bit now, and this is what we've done. I'm going to run through it. We've redesigned the hack and centromere. We've found a way to assemble it, and we've also made a way to deliver really large hacks, so megabases in size. So I'm going to run through our design of the centromere. Centromeres are really repetitive, and this has been a big problem in terms of avoiding recombination or rearrangement, and we wanted to minimize this as much as possible. We went back to this consensus sequence back in 1987, and we removed as much of the repeats as possible, keeping only conserved motifs and inserting lag oocytes. So in this case, you can see that many of the nucleotides can be varied. So the longest stretch of any repetition is actually just that lag O repeat. And we've made it so that the lag O repeats will recruit a fusion protein of any type. CNPA is one option, but there are many different proteins in the human cell that can help to establish centromere function. And this is our hack design. So it's built on a yak, because again, we wanted to be able to shuttle between bacteria, yeast, and human cells, and we want to build it in yeast, because yeast is really good at assembling really large and complex sequences. So again, it's been a yak, and as you can see, the yak can be linearized by cutting in the middle of a His gene. This means that there's the option of making your hack linear. We also have selectable markers like GFP and a drug resistance gene that is also an origin of replication, as well as the centromere, which, as I explained, has a way to self-perpetuate. Insulators prevent reorganization of your chromosome, and also human telomeres. So we're in the process of putting in the telomeres, but right now we're already testing a design with the centromere in it. It's currently only 30 kb in size, which is pretty shocking for a lot of people, but there are studies that have shown that you can work with much less. So a centromere as big as 5 kb in human cells is capable of being a centromere. And the way we've done it through assembly has been quite complicated. It took us about a year to make this hack because of all the repeats and the large centromere and telomeres again. But the best way to do it, in our experience, is through tire assembly, which is to throw all your fragments into yeast with overlapping ends. And this works on the scale that's much better than any in vitro method that is out there right now. Lastly, I just want to talk about some work that we've done to show that we can deliver megabases of DNA from yeast into mammalian cells. And this is a really big barrier, because you can't do this just through lipofectamine. You expose your DNA to too much damage in the process. But this means that you can now start creating hacks of megabases in yeast and pushing it into human cells. And you can get this at a rate of 1 in 300 cells would get the hack when exposed to the yeast. So a nice thing about it too is that if you have yeast that have your hack, you can deliver proteins too. And my collaborators managed to give human cells herpes through this method. So they assembled the herpes genome in yeast and also delivered it with proteins into human cells, and they've shown that the cells get herpes. So this is a super non-fun way to get herpes. So right now, I'm gonna run through it a little, but the key ingredient here is arresting the cells in mitosis, because this removes the nuclear membrane, and it makes it easier for your hack to get into the human cell and get accepted into the nucleus when it reassembles. And so again, the rates, if you compare 12 kb hack out to a 1 megabase hack, there's virtually no difference. They're all about 1 in 300, or like 1 in 1,000 cells will get your hack. And this is super amazing because, again, you can make so much with this. And so that concludes my talk. I just want to say that we're testing the hack now, and this work has mostly been funded by DARPA and has been a collaboration between my lab, Pam, and John Glass at JCVI. David's been working on the yeast to mammalian cell fusion technique, and Jeff has been really instructive in the design of the hack as well. And I have recently got a master's student, so I'm not the only one building the hack and testing in the lab. And yeah, these are my acknowledgements. Yep. Thank you. Question?
First of all, that was a marvelously clear presentation.
Thank you.
Including the slides, they were great.
Thanks.
I wanted to ask which different types of mammalian cells have you done this in?
Mainly we're testing in hack first, just because it's so easy to transfect, and we have protocols for the yeast to mammalian cell fusion optimized for hack cells, viral cells, even mosquito cells. So the protocol is really easy, and you can achieve rates of 1 in 1,000 delivery. But first we're doing hack cells, but we'll definitely branch out into other ones once we troubleshoot the hack. So I don't expect it to be an overnight miracle. It's gonna need some work. Yeah.
Normally, formation de novo kinetochore or human artificial chromosome is accompanied by integration into chromosome. So only a fraction of cells contain HAC if you use highly competent alpha satellite DNA, and even loading of SNP protein, for example, loading factors. Did you observe integration of your cassette into chromosome?
So we haven't gotten that far yet. Up till today, we've just transformed it in, so it's been a week since then. I haven't been able to check whether they've integrated into chromosome yet. So right now, we're just trying to check if it can perpetuate for a couple of weeks first. But that's a really good concern, because you don't want your hacks to just randomly insert into your genome. Yep.
Did you say that there's a sorry, I'm Leslie Mitchell. Did you say there's a 5 kb centromere that's functional? Did I hear that correctly?
No. So we've just made our 5 kb centromere. You mean other studies? Yeah. So what people have done is that they've made about 5 to 10 kbs of lag-all repeats and thrown that into different human cells and tried recruiting different proteins through like a SNPA or HGP, other centromere fragments, and they found that it can create a centromere, form a kinetochore.
So then there's like multimerization of that 5 kb.
They are not sure whether there's multimerization. So again, this is a huge problem about whether you get repeats or not. But as far as I know, they just insert it, in an arm of an existing chromosome, and they show that it forms kinetochore at that site. Yeah.
I'm going to talk some of the work we've been doing on microbiome engineering and how that might be relevant to some of the efforts that are going on here. And also at the end to sort of discuss about some other aspects of, if you were to re-encode a human cell or another large animal, what sort of things you might want to put into it. So I'll start off, we're really interested in finding new ways of modulating the human microbiome. As you know, it's over the last 5-10 years, there's been a lot of new insights into how the microbes that live on us or in us affect human health and physiology. But I think the tools that we have to sort of manipulate the microbiome could be much improved in terms of the precision in which you make these modifications. So right now, if you want to modify the microbiome, you might take an antibiotic, which targets a broad spectrum of bacteria, and it's not particularly targeted to any given strain or species that you might want to try and affect. And similarly, if you want to try to do a whole microbiome transplant, you can do that through fecal transplant strategies. But again, that's sort of trying to replace the entire microbiome that's already in you with something completely different. So we're pretty interested in sort of synthetic biology approaches to try to develop more precise additive strategies. So can you basically systematically domesticate all of the strains that are basically living inside of you? And could you then potentially engineer them so that they perform certain functions or record what's going on inside of your specific microbiome? And similarly, in addition to the sort of additive stuff, you want to add stuff to the microbiome, what if we could have targeted ways of removing specific members of the microbiome? So instead of trying to apply an antibiotic that kills a lot of things, what if you could remove that one or two strains or species that you're interested in targeting? So we've been exploring a couple of these different technologies. Because of time, I'll just briefly touch upon some of the things we've been doing. One of the envisioned applications of this is one day we'd be very interested in being able to take and engineer either a probiotic strain or commensal strain of bacteria that can then be delivered to a specific site, whether it's in the oral cavity, in your gut, on your skin, et cetera. Imagine that you could do this, but you could re-engineer that bacteria so that it actually contains sensors that actually detect things that you care about. Do you have inflammation? Do you have early signs of cancer, et cetera? And one application could be that this is a diagnostic, and when the thing comes out the other end, you can basically see if it changed color or whatever as a readout of what happened in your body. Alternatively, you can imagine sort of engineering these microbes to not only sense disease, but try to affect disease. So there have already been early efforts to try to engineer lactococcus, for example, or lactobacillus to express certain cytokines. And you can imagine that in the gut to treat things like inflammatory bowel disease or allergies or other sort of gut-related conditions, that you could engineer therapeutic production sort of in your gut. So to enable this sort of approach, we've been really interested in trying to load these bacteria with sort of circuits that will allow you to basically take any arbitrary set of inputs, connect them through the circuitry, and program the cell to have a specific function. So I won't go through the details on these, but we and others in the community have built a variety of different methods. For example, can you do logic? Can you compute inside of a bacterial system? Can you engineer cells to remember what they've seen from many hours or days ago? And also more analog sort of approaches. So instead of just a cell that remembers, did I see this particular signal or not, maybe you want a cell that tells you how much you've seen that signal for, how long you've seen that particular signal for. Where we're going with this is ultimately we like to enable sort of more complex cellular programs. We've started off in bacterial systems. We want programs that we can define. These are genetic programs that have time dependence, order dependence, state dependence. And we think that that'll be a powerful way of sort of programming what these cells do. At the end, I'll touch upon, I think we have a similar opportunity to do this in human cells now. Right? We're not limited to doing this in bacterial systems. And for a lot of the applications we heard about earlier, studying cell differentiation or studying what cells are doing in situ in an animal, it would be very useful to outfit these engineered mammalian cells or human cells to be able to sort of report on what they're encountering inside of a complex animal model. But for now, I'll tell you about what we've been doing on the bacterial side. Our lab has been really interested, along with Chris Voigt and other people, in trying to domesticate organisms that live on us and in us. Many of the organisms that live on us are not particularly easy to grow, although people are making significant progress in terms of high throughput ways of getting these microbes to even just grow. But the secondary challenge, we think, is that once sort of you have ability to grow these organisms, how do you start engineering them? We want to systematically try to develop tools, basically make it as easy to engineer as, for example, E. coli. So to start off, we picked the Bacteroides for a variety of different reasons. They are found in many people at very abundant levels. About 50% of the people in this room probably have B theta in their gut at sort of one of the dominant organisms. But the tools aren't great for engineering these particular organisms. So I think I won't go into the specific details of all this, but you can, using tools like CRISPR and other methods, really sort of systematically go through and build libraries of parts and sort of systematically optimize them so that you can start manipulating these systems. We put in CRISPR-I, we put in 4 different inducible systems. We built libraries of promoters and ribosome binding sites, and I think you could systematically do this for all the other organisms potentially that live on us that we care about. We've used this to sort of enable memory inside of the cell. So similar to what we did in E. coli, we use recombinases here in the upper left corner. They basically flip pieces of DNA, and so therefore you can use them to sort of encode memory in the DNA. So we've used this, and we've shown that you can actually integrate cassettes. Here we have, I think, 10 or 11 recombinase sites integrated into the genome of B theta, and we can use these recombinases to basically store information of what that particular organism has encountered. You can use these, you can colonize in the future people with these, but now we're doing this in mice. We colonize mice with this particular organism, show that you could feed the mouse specific food sources like arabinogalactan and actually turn on gene expression in the gut of the mouse. So this is looking at a mouse that got fed arabinogalactan, and you turn on basically luciferase production in the gut. And when you remove it, which is at the end of this gray bar, it goes away. Similarly, we've implemented CRISPR-I sort of ways of turning things off using this particular approach. So just showing that you can sort of do remote control, and other folks, for example, Pam Silver's lab, show that you can also do remote control of sort of what organisms are doing in situ using this type of approach. So what are some of the things we've been thinking about that might be synergistic with this particular program? Could you systematically domesticate all human microbes? Could you then really start looking at not just single bug interactions with the host, but sort of looking at combinations? And I think the combinations are where there's a lot of interesting things that will arise from this. Maybe you do that in vitro first and ultimately go to an animal model to do that. Can you then take advantage of this intermittent interaction between the host and these microbes to really engineer therapeutic or diagnostic interventions? We think some of the memory and computing circuits that we're developing can be useful for really studying what these bacteria are actually encountering in situ. And then sort of thinking forward, I think the tools that we're developing in this field are not just limited to bacterial systems. We can start thinking about putting them in human systems. And sort of we heard ideas earlier about sort of universal cells or safe cells. One of the things that we're quite interested in is actually putting genetic recorders, give me 1 minute, like genetic recorders and circuits into these cells so that we can actually study what they're doing now in a mammalian context. And we heard a little bit about this earlier. I think one of the grand challenges of really why maybe you want to do HGP2 is you really want to systematically start to unravel what are these combinatorial interactions, what are these complex interactions that we can't really just get by observational studies alone. So in the remaining 30 seconds, the other thing that our lab is really excited about is really trying to basically build measurement devices that go into complex mammalian systems. So this is a complex Intel chip, and you can basically take a chip like this, use a probe station, actually study what little transistors are doing, and ablate them and change them. We don't have similar things necessarily that are easy to could you go into any cell in a mouse, ask that cell what it saw over the period of that experiment? You can't really do that easily. So what if we could actually put in genetic recording circuits into every single cell in this animal, and basically have them self-report or record what's going on throughout the lifetime of that experiment. And then you could collect them later and actually see what happened to those specific cells in situ. So we're not just limiting ourselves to sort of in vitro biology. I think the challenge this might help us solve is really understanding what's going on in a complex multicellular system, but also helping us to potentially unravel some of these combinatorial approaches. And so I think in the remaining time, sort of this is the idea. Can you engineer cells that sort of remember what they've seen, use DNA as a recorder, and you can imagine other sort of recording devices, but if you could record stuff in DNA, that might be very powerful as a readout. And then at the end, we're also very interested in sort of enacting and also understanding the order dependence of these activities. So we've started building things like state machines where these are genetic circuits that have different unique sequences. And depending on what order of events, so maybe they see Wnt first and followed by some other growth factor, or the other way around, AB versus BA, this will differentiate these circuits actually into unique DNA states that you can then just read out later on. So imagine being able to do this, plug this into some engineered stem cell or embryo or whatever, let it develop into a mouse, and then basically collect the cell types you care about and read out what, push that cell down to that location. So we've built these systems in bacteria. We think it's going to be useful for mammalian systems. These are highly scalable. You can take 3 inputs, for example, and encode 16 states. And we've done some mathematical computations to show that basically with n inputs, you can encode 3 to n different states, at least with this given architecture. So with that, sorry I went over time, but that's what we're interested in doing. Thanks. Yes?
Cool talk. I'm just curious if you could also try and measure or even encode some of the epigenetic states, like in E. coli, you can stratify based on methyl-6-adenosine, or record what happens with phage interactions.
Right. No, I think with any of these genetic recording devices, as long as you can couple whatever you want to record into some gene regulation that controls the expression of the recombinase or the things like the CRISPR system or whatever, you should be able to record that. Yeah.
Hi, everybody. I'm Amy Wagers. So we've been talking all morning about ways and situations in which one would want to recode or rewrite the human genome. And this is an interest in my laboratory with particular respect to overcoming inherited mutations that give rise to devastating human diseases. And one of the ones, sorry, this is echoing a little. One of the ones that we're particularly interested in is Duchenne muscular dystrophy. This is an X-linked disease, inherited disease that affects about 1 in every 3,000 to 5,000 boys. It's a progressive and devastating muscle degenerative disorder for which there is no cure and really no effective therapies. DMD arises due to frame-shifting mutations that lead to loss of function of a protein called dystrophin, which normally serves to link the internal cytoskeleton of skeletal muscle fibers to the extracellular matrix in order to provide them with structural stability so they don't rip apart when they contract. So mutations, as I mentioned, that cause Duchenne muscular dystrophy and lead to this severe, progressive, and deadly disease disrupt the normal reading frame of the DMD gene, which is encoded in a very large amount of DNA. It's 79 exons and comprises actually a full % of the X chromosome. When these mutations occur, these can include insertions, deletions, point mutations that disrupt the reading frame. This destabilizes the mRNA, and you have no protein product. What's interesting, however, is a related class of patients who have a form of muscular dystrophy known as Becker muscular dystrophy, which is much more severe. So while the expected lifespan of a DMD patient is 20 to 30, many Becker patients can live a full lifespan, and actually some even remain ambulatory into their 60s. And the reason for this is that the mutations that give rise to Becker muscular dystrophy, while they occur in the same gene, actually don't disrupt reading frame. They lead to internal deletions that maintain reading frames, so you get a partially functional protein with a sort of shortened middle that subserves many of the functions of the normal dystrophin protein. And so this has led to the suggestion that if one could recode the DMD mutations such that they remove the frame-shifting mutations or remove exons adjacent to frame-shifting mutations in order to restore reading frame, this could convert a patient with a very severe muscle disease to a patient with a much more mild muscle disease. And this is being investigated already in a pharmacological strategy using antisense oligonucleotides to disrupt splice donor and acceptor sites of particular exons so that the mRNA level one includes or excludes particular exons of DMD. And in addition has been used to engineer sort of synthetic dystrophin constructs that can be delivered, so-called microdystrophins, in place of the mutated gene. But our lab was interested in taking an approach of actually recoding the gene itself by removing exons that carried frame-shifting mutations and restoring the frame of dystrophin within endogenous muscle fibers and their progenitors. And so we attacked this in the mouse as a model, the MDX mouse as a model of human Duchenne muscular dystrophy. It has a point mutation in exon 23 that disrupts the reading frame and leads normally to nonsense-mediated decay of the mRNA. But if you suppress inclusion of exon 23, this leads to splicing of the 22 and 24 exons and production of an in-frame but a partially truncated protein. And so using CRISPR-Cas9 technology, we wanted to see whether we could design a strategy that would lead to excision of this exon and production of a hybrid. And we did this by packaging CRISPR-Cas9 into AAV, showed that it was possible to deliver this locally into skeletal muscle by injection of a dual system with one adeno-associated virus encoding the Cas9 protein and the other encoding guide RNAs that target it to the intronic sequences. This leads to excision of the exon 23 sequence, production of a truncated mRNA, and restoration of dystrophin protein by Western blotted immunofluorescence. And very importantly, it restores contractile function in the skeletal muscle where this gene is edited and it protects from damage. This strategy can also be used systemically, not only locally, by injection into the bloodstream of neonatal mice, as shown here, and you get editing frequencies up to 15% within different muscles, including the heart, which is extremely affected in DMD patients. And in adult animals, while the frequencies are variable, you do get production of dystrophin protein that's restored in both skeletal and cardiac muscle. And then very importantly, because restoration of or editing of the gene in the mature muscle fibers is therapeutic but may not be maintained if the progenitor cells that seed replacement of fibers aren't also modified, we developed a reporter system as well as a targeted system against the DMD gene itself, and showed that the endogenous progenitors, the muscle stem cells for turnover of skeletal muscle, are also edited, albeit at relatively low frequencies. We've not been able yet to get over 5%. But this is sufficient to give rise to colonies of muscle cells that actually re-express dystrophin protein and have a sort of ameliorated phenotype. So in conclusion, what this strategy allows one to do is target disease-causing mutations, in particular affected tissues as well as their progenitors, providing restored protein expression, as well as the capacity of replacing that restored gene through progenitor regeneration of muscle fibers. And I think this is interesting because it gives us the possibility of converting a very severe disease allele to a more mild allele, and even an allele that normally occurs within the human genetic gene pool. So with that, I'm happy to answer questions. This work was done in my lab together and spearheaded really by an outstanding PhD student, Sharif Bordbar, with help from Chris Zhu, Jason Cheng, who's an undergraduate, and 2 technicians in the lab. And we collaborated with Feng Zheng's lab, Jeff Woodrick on muscle physiology, Luke Vandenberg on the AAV, and George Church and Wei-Long Chu in his lab. Thank you very much.
Just a question. Was 15% therapeutic? Did the mice become normal?
So the mice will never become normal in this strategy, and I think that's an interesting aspect to discuss. So we're taking a very severe disease allele, and turning it into a less severe disease allele, but they're never going to be normal in the sense that they don't have a complete restoration of the coding sequence of the gene. In patients, it's been estimated that you need to achieve about 25 to 30% expression of dystrophin protein in order to be relatively asymptomatic, but you have a restoration of function with smaller amounts. And that's, of course, a challenging estimate because you have regional editing. So every aspect of the muscle is not equivalently edited, and so you might have variegation of the restoration of the phenotype. Yeah?
So why not just actually edit the allele to be the correct allele?
Right.
Chop it out.
Right. So obviously the long-term goal would be to perfectly correct the gene. In order to, so the first important aspect of that is the gene is very large and there are very many different mutations. So you probably need to design particular strategies for individual patients. Some of the exon excision strategies could cover up to 15% of patients with a single approach, which has its attractiveness in terms of the regulatory path. And as a first step. The other issue is that to fully correct, you probably need to enable homologous recombination to occur in multinucleated postmitotic cells, which is a challenge. And one of the reasons we're very excited about the fact that we get even low levels of editing in the progenitor cells, which can be proliferative and might support complete repair, and that's something that we're working towards.
Hello, I'm Yinan Khan from the Church Lab. My question is, your method requires both guide RNAs to work in vitro to splice out exon 23.
Yeah.
Can you use one guide RNA to target either a splice acceptor or a branch point of that exon and to achieve the same effect?
Yeah, so in principle, yes, you could use a single target site. In this particular case, there was very high homology between the splice acceptor sites in multiple exons, and so for specific targeting, it was more challenging. And then a nice thing about targeting the intronic sequences is, of course, you're tolerant to various indels that occur in that region because you're creating this hybrid intron anyway. Using some of the newer nucleases that generally create larger deletions, one might be able to come up with strategies that would only depend on one guide and not two. But I will say that in the couple of experiments that we've done looking at that, the efficiency of excision is not suppressed by the need for two guide RNAs. Thank you.
I'm going to talk about a project we conducted that I believe will just about half the work you need to do to write the human genome. So that's great. I mean, essentially we already did half of the work, so why not do the rest of it? In which one? Okay. So human eukaryotic genomes can exist in various ploidies. And there's a reason why Jeff chose yeast and the haploid form of yeast to make a synthetic chromosome. I guess if you want to write a genome, you would rather do it once than 8 times, such as in a strawberry, or corn was also mentioned earlier. So I think haploid has a great advantage. And the reason is not just because it has less DNA in it, it also greatly facilitates the analysis of the genome. And I think there's great value of doing this in human cells, because ultimately if we want to understand the human genome, we need to be able to change it as much as we can and to gain the most functional insight. Haploid cells, there's of course an absence of complementation by another allele, so whatever you do to the cell should be functionally expressed. Haploid cells are usually just in the germ cells, and these cells do not divide. Neither sperm nor eggs make more eggs or sperm. So the question really was, can haploid human genomes divide? And if a cell that is just haploid, can it also differentiate? So what we did in my lab in New York is we took oocytes, human oocytes, and they have, when they are at meiosis II, they have a replicated genome, sister chromatids, and then they segregate those into a polar body. So they just segregate sister chromatids. So the oocyte is already haploid. But then you have a single unreplicated genome at the one cell stage. These are the polar bodies from the meiotic divisions. And then you start development as a haploid. These cells start development as a haploid cell. Occasionally, the cell does not divide, and then you have 2 nuclei in one cell, which generates a diploid cell. So you end up with cells, a mixture of cells, when you derive a cell line that's both haploid and diploid cells in there. So what we had to do, and this was done in collaboration with the lab of Nissim Ben-Venisti at Hebrew University, is to isolate those cells. And this can be done simply by staining those cells with Hoechst and then analyzing them in a fax and sorting them. And you can see here, the more intense the Hoechst staining is, the more DNA it has. The less intense it is, the less DNA it has. And so haploid cells in G1, the unreplicated ones, they would be unique. There is no diploid cell that has as little DNA as a haploid unreplicated cell. And this way we can generate highly pure cells that are haploid. You see that here, and this would be the karyotype. What these cells do is they can be highly enriched, but over time cells do diploidize. So when a cell fails cytokinesis, and even it's just a fraction of the culture, because there's no reversion to haploidy, the cell culture gradually becomes more and more diploid, so sorting needs to be repeated perhaps every 3 to 4 weeks. The karyotypes are stable. They don't have specific deletions or duplications that would make them pseudodiploid. They're truly haploid. And there are easy ways to check for their ploidy. One that I really like is simply do an immunostaining, and you check the number of centromere spots. That works well in ES cells. It doesn't work so well in differentiated cells. And if the number of those is around 23, it's a haploid nucleus. If it's around 40 or to 46, it's a diploid nucleus. You can also do FISH, but this is a technique that's established in every single lab, and it's very easy to do. And so is sorting. It's not hard to purify those cells. There's only very few differences between haploid cells and diploid cells, surprisingly. They cluster in terms of gene expression, but of course they have one X chromosome. In human embryonic stem cells, they often inactivate one X, and that means the ratio of X to autosome, which is what shows here, that would be the exome in gene expression, is 1 to 1, 1 to 2, so 2 autosomes to 1 X chromosome in those X-inactivated diploid cells, while haploid cells have an active X chromosome, and the ratio is 1 to 1. So relative to the autosomes, the X chromosome is slightly overexpressed. But this doesn't seem to affect those cells in a remarkable way. They're also smaller. They have a smaller volume, and they have a less total RNA. We know that they can be transfected. They can be used for genetic modification. We did proof of principle genetic screen on that. This just shows that they are amenable to genetic manipulation. And the question we then had, can they differentiate into various cell types? Various eukaryotic organisms can exist as haploids, as listed here, but we didn't know for human cells what they're capable for. And what you see here, we differentiated those cells into various lineages and cell types. Here you see neurons. Again, they're haploid. This would be the FACS sort. The majority of the population is haploid. Here, the centromere staining. Then the same for cardiomyocytes. And the same for endodermal cells. We also made some beta cells, not shown here. But these are pancreatic progenitor cells, and they too haploid. Again, there's a trend to more cells becoming diploid, but the significant fraction remains haploid. What you can see here is a so-called teratoma, which is the in vivo differentiation of these stem cells. And you can see that a surprisingly high number in these highly differentiated structure is actually haploid here, fish, and FACS analysis. So human cells as haploids have the capability to give rise to various differentiated cell types. What is the strengths of this system? As I mentioned, it allows us stringent molecular and functional testing of any genetic change. It can be differentiated in any cell type of interest. So my research over many years has been the target of many ethical studies, and the discussion we've had before reminded me very much of this. And I think what often happens is that there is misinformation that leads to restrictions on research rather than on strong regulation how this is used in the actual application. And I couldn't agree more on this, Scott, that I think the focus should be on what is done with it and not holding back on what research we pursue to determine whether there is value of this and what it could be ultimately be used for. And this is a law that, as of today, still affects stem cell research tremendously, in particular this. The NIH could not be involved in this work, and it is because a misinformed law equates a human embryo with parthenogenesis. These cells that you've just seen, they have no potential to make a human being, and yet they fall under that same law, unfortunately. If asked the NIH whether they would reconsider including those cells into the registry, the answer is no. So these are the people who contributed, mostly like Nissim Benvenisti, very good collaborator at Hebrew University, and funding was partially provided by the New York Stem Cell Foundation. Thank you.
I was wondering, this is Liam Holt again. I was wondering if you had tried to fuse two haploid cells together, two distinct haploid cells.
So, well, I mean, these are parthenogenetic ES cells, and then, you know, you're trying to make endogenetic ES cells as well, and if you fuse them, you would have a diploid biparental ES cell, for example. Could be done. We haven't done that yet, but right, I think the focus here is on keeping them haploid. There's many things that could be done with those cells, clearly.
Yeah, Jeff Buka, I was wondering, practical question. You said you have to sort them every so often.
Yes.
What's the mutagenic impact of one round of sorting on the genome integrity? In other words, how many mutations do you accumulate every time you do a cell sorting experiment?
You know, it's a good question. We don't know the answer yet. I mean, there's a Hoechst stain and there is a low dose of UV. I suspect there is some impact, but we haven't quantified it yet. And it looks as if on the level of using those cells, it certainly hasn't affected their function or their ability to grow and differentiate. But Hoechst in and of itself and the low dose of UV is probably not that toxic. It's regularly used in animal cloning. But I think we have to address that question.
What's the mechanism for diploidization of these cells?
We don't know yet the answer to that, but I suspect it's a simple lack of cytokinesis in a fraction of the cells. If you grow a diploid ESL culture, you will have a portion of tetraploid cells. These cells don't really become diploid anymore, but because they have a supernumerary centrosome, they often divide abnormally at the next cell division. So you don't really convert your entire culture from diploid to tetraploid. In this case, if you would culture for a year without sorting, you would probably lose most or all haploid cells.